switch on the pointer already. Yeah.
Okay, so our next speaker is Thomas Gröger, that will not present about food, I think. Nope. So he will be speaking about the application of comprehensive two-dimensional gas chromatography for pharmaceutical application investigation of complex drugs. Thomas.
Thanks a lot for the introduction. And as you already mentioned, we are moving a little bit away from the food analytics to another field of application, to the analytics of complex drugs. So I have to say I'm not the first author of this study, because also work was done by our PhD students, Lukas and Ole. Lukas is also in the auditorium. And we have here also a strong cooperation partner. It's a pharmaceutical company in Germany. And that's also the reason I have to put this message here, preliminary data, R&D research. Okay, let's go on. So when we handed in this talk, we thought, okay, it's maybe not of a broad interest. But then we saw the agenda which you put together, and there you see there seems to be a broader interest to use GCxGC method or other multidimensional methods for the analytics of pharmaceuticals. Because you have here biopharma regulation. We had yesterday the keynote lecture, regulated environment, which I was very surprised. But we had also some other stuff, for example here is the analytics of chiral pharmaceuticals, aspirin, ibuprofen, for example. But also this topic we have seen a minute ago, the xenobiotics. This is a raw material for pharmaceutical analytics. Roughly, you can classify this application here according to the active pharmaceutical ingredients. So the drugs there are the small, so it's a classification, it's a rough definition. So you have the small molecules like aspirin and ibuprofen. They are very well characterized. So you have one compound and you know what it is. Then you have a little bit more diverse class, for example, the biologically complex drugs, xenobiotic, for example, also the traditional Chinese medicine, but also the antibiotics, what we have seen in this study, we can classify to biologic drugs. There are other applications. You can have a look at a very recent review here from 2021, I guess. And you see there are other fields of interest, but I would like to go away a little bit from this one because we have a third class of drugs, which is not so well known, but it is a very complex drug and it is approaching very, very fast. So you have this class of non-biological complex drugs. They are very diverse in physical properties, but also in chemical properties. And they are not one compound, it could be thousands of compounds which make it. And when you look for it, because they are so diverse, they are not specified what they are. They are specified what they are not. So basically, it is not a biological medical product, or it's not biological drug. It is not an homomolecular, but continuous different structure, so it's very diverse. And what is very surprising for me as an analytical chemist, you cannot fully characterize it by a physical chemical method. And this was, of course, for us a thing, okay, should we accept this challenge? And there is another thing which is very important for us. Since you have not the full identity of the product, it is defined by the production rate. And this is very well controlled, of course. Now, having a closer look. So on the market, there are some products already, and they are very old products. They are approved since decades. So this product since 1800 something, for example, it's on the market. And of course, the production is, the way of production is very defined. You have approved it by the local authorities. So maybe you have approved it in Germany, in France, and so on, but not maybe in Italy or something. And of course, the effect is also proved. What is now the problem now with this compound? The problem is now the new regulation which are popping up. Because as soon as you're touching this product, for example, you would like to do a further new registration in Italy or something, they would, the European Union says, or the European regulation says, now we would like to have the detailed information. The second thing is, even if you would like to make a follow-on project, product, which is maybe not so different, a little bit different in composition. They ask you, hey, now we need the detailed description. And even if you improve the production process, they say, no, your product is approved according to the known production process. Now we need this new detailed characterization. And if a company which has such a, we can tentatively assign it to an MDCW, it's a sodium bitumen sulfonate. It is, as mentioned, approved according to a well-controlled manufacturing process. It's a European product, you can say, because the first part, which is not regulated, is done in France. It's a non-biological product. So you have a rock here, for example, where you do a dry distillation, and what you get out is a shale oil. In shale oil, we are familiar. So everybody who is doing petrochemistry, you can say, shale oil, we can do this. Then you have some steps which are under GMP, which are done in Austria, for example. You have a vacuum distillation, cleaning up process, raffination process, then a sulfonation. So you have already much of sulfur in the sample, and then you add a sulfonation process, then neutralization, then you have the active pharmaceutical ingredient, and then the final formulation is done in Germany, and then you get out all the products. So when we would like to analyze it now, what are the requirements for analytical technique? So from the textbook, we would say it's not possible because, per definition, it's not possible to do it. So point out something. What we need is, or what was our aim at least, we have thousands of different compounds because we know a little bit about the product already before, because it was this crude oil was a little bit similar to crude oil. So we would like to know for every compound is an elemental composition. This is at least the basics. But when you are a little bit familiar with MS instrumentation, sulfur contents are a little bit tricky there. Then the next step would be an isomerical or structural composition. Okay, these are more qualitative aspects, but we have also a quantitative aspect because we have this well-controlled, robust manufacturing process. And that means we have also to detect these variations in the manufacturing process. So we have to deal with those. And what we tried first, very brief, is our liquid Pegasus system. It's now almost or more than 10 years old. But with all these modifications, it's still running very, very robust, I have to say. But I will not go into detail at the moment about this. I will directly go into the analytics, and we will start with the distillate, which is already in the GMP. And for this distillate, we were not very surprised because distillate petrochemistry, we know it should look like this, which is a little bit, difficult is again the sulfur content because you have to deal with it. But in generally, the HI-TOF system with a GCxGC, you can all make the nice things what we have seen before. The classification, for example, you can apply here, of course, a Legion order. We have the accurate mass here, where we can do this elemental composition calculation. And also you would have also the isotopic pattern, and of course, you have the fragment. So this is not a big deal, but it's a very important first step. But this is not the API, of course. This is a precursor compound. The next step was, of course, to deal with also this sulfur content, because the high resolution, we have a high resolution system, but it's only going up to 50,000. But Lukas did a very nice study. Normally, compound which has a sulfur inside and isobaric compound, and also only the pure hydrocarbon, will not overlap in your chromatograph. So if you have this high separation power, this on high res system, and then you can also deal with this. And with this, you can do everything what you are used to do with this matrix. The distillate, very nice. All the compound classes, and also for the sulfur compounds, and also for the other heteroatoms, for example, phenols and pyridines. So the challenge for the distillate was not very high. The next step is the manufacturing process, comparing, for example, the distillate and the refined distillate. Refination means they add a little bit acid, they add a little bit inorganic salts to clear the composition here. But on a first glance, you don't see so much differences here between. And for that, we have to do a quantitative analytics, and you can do this either individually, tile-based, for example. And I moved out all the slides about tile because I think now you know what is tile and how it works. And I only would like to show you results here from the classification. And if you would like to do this to find out the minor differences in the production process, very important here is statistics. So we do it a bit first a group test to an outlier test. And you see basic message is everything is green, it's fine. So not only the qualitative aspect is okay, also the quantitative aspect is okay by applying GCxGC. So we have a little bit more variances here for this compound classes, but this is mainly the abundance. They were only a little bit in the compounds, sometimes below the detection limit, sometimes above the detection limit, and this we can classify. What we saw already, see now is the T-tech. And there we saw a little bit differences before and after, but these differences are very important for us because these are the wanted differences. Because what you can see, you don't affect a lot the whole composition of the matrix. But the manufacturing process and this raffination is removing some harmful compounds which are regulated. For example, phenols and here pyridine, for example, this enhancement factors 30. And this was exactly what we have to monitor. So also I would say the quantitative aspects here are solved, but this is only the distillate and the refined precursor. We are not talking at the moment about the product, the API, because the API is sulfonated, and now it becomes tricky because a sulfonated salt, what we get out, you can't analyze with the GCxGC system, or it's very hard. So the next step is to think about how can we analyze it. Of course, you can do derivatization here to decrease the boiling point and the polarity, and this is maybe acceptable for GCxGC. So we did an analytics with this, and you see it is now possible to deal with this, but this is now a really unknown matrix because sulfonated products you won't find in the literature. You are even not able to buy it very much in the standard substances, but you know what is the precursor. You do a sulfonation. This is the first hint for the analytics. Then you have very limited database entries, maybe for the products in the beginning. You have, unfortunately, only standards for 2 or 3 of the products, not very much. But again, the starting material is known. And of course, elution profile, fragmentation pattern, and what we have is the elemental composition. And this was the reason that we were able to make also for the API a complete entire qualitative analytics at least. And what you see here is sort of compounds, which are the composition of it. And it was the first time they're dealing with these problems now, I think since 30 or 40 years already. And it was the first time we were really able to identify for the class and what is inside. Okay, now we can say, okay, let's stop the presentation, job is done. Unfortunately, it's not true because we have now applied for an unknown matrix, an unknown method. And when you do GCxGC, you often say, okay, I have this amount of data, thousands of peaks. This must be my matrix. But unfortunately, this is not true because you have your data are the matrix, but also the analytical technique you applied, the sample preparation you did, and of course the operator who is handling it. But now have we a problem how to validate such non-targeted data when you don't have an appropriate reference method. And this is a big problem. So a conventional way will not work because on conventional way you have normally one target compound, you have maybe a deuterated compound, you can make recoveries. For a non-targeted approach, it becomes a little bit harder because this traditional way will not work. So we decided we do some risk assessment. So you look to your method, make a little bit brainstorming, prove it a little bit, and then you put together everything what could have an influence. Then you say, okay, it's an expert judgment. Some of these aspects we can handle. We are experts, so we don't have to take care. And based on this, you do it. You apply everything to everything of this at risk level, low, medium, high, for example. And for this, we have a high risk level. Of course, you have to look a little bit closer because they will affect your your data. And for this, we put out 3. We have looked to more of them, but 3 I think are very important when you do GC, for example, GCxGC, because they will affect or discriminate compounds. Of course, you have the sample preparation. If you do an extraction, maybe not everything is extracted. The second thing is a hot injection. Most people oversee in GC, you put 200 degrees, 300 degrees to your sample. This maybe there's some decomposition. And last thing is, of course, maybe your method is completely incompatible with the physical chemical property. So we have a look for this one, very, very brief, because we have not so much time here. Sample preparation. So Lukas spent a lot of work to compare different extraction methods. Solid phase extraction, also liquid-liquid extraction, but also direct injection with derivatization. And you will see you have different. So these are different compound classes here, and these are the carbon number of these compound classes. And you see what you see in your chromatogram is a little bit different depending what you are doing. But what was very important for us, we are not discriminating a complete compound class. Maybe a little bit the carbon numbers, but not the complete compound. And this we did for the extraction methods, but also for the derivatization. So we have now a little bit insight what we are seeing and what we might oversee. The second thing we also checked was the thermal injection. And this is always a big issue, I have to say, because normally you don't care about. You inject, you see peaks, hey, this is my matrix. But here we have you have seen this complex instrument what we are had in the beginning, and here we can also do some thermal analysis with a thermal balance. Thermal balance is very easy. You put your sample on a balance, apply heat. You can go up to 1000 degrees, and then you have evolved gases in which you can feed directly to a high resolution. What can we observe there is if we have some decomposition. First of all, we see when it starts to evaporate, and with the NF fragment, we can also see is it more decomposition. If we see a lot of CO2, for example, or a lot of unsaturated compounds, then it most likely break down products. This we did with the limited number of standards we had for example, sodium benzene sulfonate. You see it is evaporating at some point, but it is a decomposition. So a direct analytics would be not possible. And this we did with the derivative standard. And there you see at 250 degrees, it's going into the gas phase. And when you look to the MS, it is really no decomposition. So for the standard, you saw, okay, it's possible for the derivative. But now have a look for our sample. So it's sodium bitumen sulfonate derivative. We did this also with the intermediate step where you see a lot of decomposition. But there, when you check the chromatogram, not the chromatogram, sorry, the mass fragmentation here, so it should be the intact compound. So we can also say thermal injection is working, at least for the derivative. And the last thing we did also is now going to a completely different method, because maybe we only see a very tiny spot of our matrix. And for that, we thought, first of all, going to the liquid phase, making something without any preparation steps, so direct infusion. And this is an FDICR system, what we have in Rostock. We have the second PhD student is working with. And then we did a comparison. So complete different instrumentation, complete different physical chemical properties, and to compare it. Of course, now we have a little bit the problem because they are talking a different language. The data from FDICR looks completely different what you get from a GCxGC. You have peaks, and on the other thing, you have fragments and some spectra. What we have in both cases are high resolution mass data. because we applied a high resolution mass system. And then you can calculate the dBE double bind equivalent. I will not go into detail, but this is the language with which both talk. And now we can compare these results here from both instrumentation, and we see, hey, they are really similar. And what does it tell us? We are not losing compound classes or something else. So with our method, which we are applying, okay, we should see almost everything. We cannot say that we see everything, but it's a good hint. Okay, and with this slide, I would like to end my presentation and make some key messages. So first of all, GCxGC, we have seen, is very fast developing in the field of pharmaceutical analytics, but of course, also LC, what we have seen yesterday and the day before. And what I was really surprised also in the regulated environment, because I think our instrument we will never can put in a regulated environment, but an LC system we have already seen, and maybe a regular GCxGC system will also go forward. We have seen you maybe cannot fully characterize this physical, chemical, analytical methods by conventional methods, but try a GCxGC system, and maybe it will work. And the last point is maybe we should draw a little bit more attention or pay a little bit more attention to the validation of the method. I know it's a very elaborate, time-consuming process, and most likely your data will end up only in the supplement of your paper. But I think the system is okay. A GCxGC system is working, but this validation may be a little bit more attention for it. And this, I would like to end my presentation with the acknowledgement. Of course, the two PhD students which are doing this experiment, Lukas and Oleg, and also Sandra, which recently joined our group. She's digging a little bit into the data. If you're interested in the first part of our study, it's already published last year in ADC. The second part about the validation step and how you can validate such a complex method will be handed in, I think, hopefully in the next 2 months, and maybe somebody of you will become the reviewer for this paper. So now you know the background and all the hard work, what was there. Maybe if you're interested, we have also some posters upstairs, which are dealing not with this instrument, but with the next generation. LICO was so kind to have an analytics on an ultra high resolution system, where we can also resolve the sulfur split, but also we apply different ionization methods. I really want to thank our cooperation partner, Ichtiol, because they are pushing really the project forward, and they are really talking to us, not only give us the results and then decide. So they are really pushing this project, and they are doing a lot of research. And if you have more questions, you can come back to me, but also Lukas is present. And I would like to thank you for your